Hello, everyone. My name is Nushin Tabatabai Zavare. I'm the Associate Director of Immunology at STEMCELL Technologies and would like to discuss T-cell manufacturing using our GMP ImmunoCult reagents in closed bioreactors. In T-cell therapy, the manufacturing process is very important for the quality of final T-cell product. There are many parameters in the manufacturing process that can be adjusted to tailor your final product to your needs. We offer GMP-compliant ImmunoCult reagents, including a medium and 2 activators designed for activation and expansion of T cells, and they can be easily incorporated into your individual workflow. These reliable and high-performing reagents are GMP manufactured, and drug master files were submitted to the U.S. Food and Drug Administration. If you are interested in testing them in your own workflow, please visit our website or contact your local stem cell representative. Today, I will present data showing the performance of these products in 3 different scale-up bioreactors: G-Rex devices, Zuri Cell Expansion System W25, and PBS Mini bioreactors. First off, I start with the application of G-Rex bioreactors for scale-up in T-cell manufacturing. G-Rex devices are equipped with gas-permeable membranes for optimal oxygenation and cell growth. Here is the protocol to expand T cells using ImmunoCult reagents in G-Rex 6M well plate bioreactors with 10 square centimeter gas-permeable membranes and a media capacity of 100 ml per well. T cells were seeded at 1 or 2 million cells per square centimeter in a 10 ml volume that includes ImmunoCult XF medium, CD3, CD28, CD2 activator at 2 different doses of 6.25 and 25 microliter per ml, and IL-2. Cells were cultured for 3 days, and cultures were topped up to 100 ml with medium and IL-2 on day 3. After day 3, no culture manipulation was performed until harvest on day 10. Here is the T-cell expansion data showing cumulative fold expansion and viability on the left and cell density per square centimeter of bioreactor or surface density on the right. We observed similar viability shown in orange dots on the left graph across different seeding cell densities and activator doses tested. However, the first condition, when the lower seeding density and lower dose of activator is used, results in higher fold expansion, the first bar on the left graph, and similar or higher surface density on the right compared to other conditions. This data shows that our reagents are flexible and support optimal growth across various culture conditions, including lower dose of activator in G-Rex bioreactors. Now looking at the phenotypic profile of subsets among expanded CD8 positive T cells on the left, we see no major differences in the proportion of central memory T cells characterized as CD45 RO positive, CCR7 positive, and shown in orange among the conditions tested. Calculating the surface density of CD8 positive central memory T cells shown on the right, we saw that high numbers of these cells are generated in all conditions tested, again indicating the compatibility of ImmunoCult reagents with the G-Rex culture environment. We examined the functionality of T cells expanded using ImmunoCult in G-Rex plates. Shown here in 2 donors, T cells were able to specifically kill NOM6 target cells. We further evaluated T cell expansion in a larger device, G-Rex 50M. This system has a media fill volume of 500 ml with a surface area of 50 square centimeter. Similar protocol to 6M well plates was used, with the exception that larger volumes were used and that cells were harvested after 7 days of cultures. After 7 days of expansion, on the left, starting with 1 million cells per square centimeter and 6.25 microliter per ml activator, the culture achieved approximately 20-fold expansion with over 90% viability. Using the same condition on the right, 20 million per square centimeter or 1 billion cells in total were generated in the G-Rex 50M system. Therefore, G-Rex system is scalable and allowing you to achieve the desired number of cells. Next, I'll discuss scale-up using Zuri cell expansion system W25. This is an automated instrument that uses a rocking motion to expand T cells in cell bags. As shown in this workflow, pan T cells were first activated on day 0 with the ImmunoCult activator in ImmunoCult XF medium with IL-2 in a culture flask for 5 days. On day 5, cells were inoculated into a cell bag and expanded in ImmunoCult XF medium with IL-2 for another 5 days on Zuri. Perfusion was initiated 2 days post-inoculation, and the perfusion rate was adjusted according to cell densities. Over the duration of the culture period, we monitored various parameters, including viability, expansion, and phenotype. The graph here on the left shows the T cell growth and viability. T cells cultured with GMP ImmunoCult reagents showed robust expansions of 500-fold on day 10, as shown in the orange bars. T cells maintain high viability throughout the culture period, as shown by the orange dots in the same graph. The graph on the right shows the phenotypic profiling of the expanded CD8 positive T cells. Over the culture period, there was an initial increase in the central memory subset, shown in orange, followed by the expansion of the effector memory population, which is CD45 RO positive, CCR7 negative, and shown here in a teal color. Despite the reduced frequency of central memory T cells at day 10, the total number of central memory cells remains constant from day 7 to 10, again shown in orange line. The reduced frequency is a result of the expansion of the effector memory population. With this knowledge, you have the flexibility to select which day to end your cultures to achieve a balance between the cell yield and desired phenotypes in your final products. A large proportion of culture CD8 positive T cells produced interferon gamma and TNF alpha, shown on the left. In addition, on average, 40% of expanded CD8 positive T cells produce granzyme B, which is one of the effector molecules for target cell killing. This data suggested that T cells expanded in ImmunoCult using Zuri workflow are highly functional. Lastly, I'll focus on PBS mini bioreactors, which use a vertical wheel technology or vertical impellers to create a homogeneous low shear environment for scale-up. T cells were first activated in flasks with the combination of medium, activator, and IL-2. After 3 days, cells were inoculated to PBS mini 0.1 with 100 ml media capacity. On day 7, cells were transferred to a PBS mini 0.5, which has a 500 ml media capacity. Cells were sampled throughout the culture period and harvested on day 10 for analysis. As shown on the left, a high fold expansion of 614 and a viability of over 95% were achieved by day 10. Looking at cell subsets within CD8 positive T cells on the right, the proportion of central memory T cells shown in orange peaks on day 5. However, the absolute numbers of central memory T cells peak on day 7 and remains constant until day 10, allowing again flexibility on the harvest time point. We tested the functionality of T cells expanded in GMP ImmunoCult reagents and PBS mini vessels. As expected, CD8 positive T cells were positive for intracellular interferon gamma and TNF alpha after PMA ionomycin stimulation, shown on the left. Also, 90% of unstimulated CD8 positive T cells produced granzyme B, which is essential for their killing capability. In summary, I've shown you that our GMP ImmunoCult reagents are versatile and flexible. They support robust T cell expansion in the 3 common bioreactors for T cell manufacturing, G-Rex devices, Zuri system, and PBS mini bioreactors. We consistently achieved high cell densities and as a result, high yields of T cells in at least 2 different sizes of G-Rex bioreactors, 6 ml plates and 50 m system. Both Zuri system and PBS mini bioreactors generated high total fold expansions and high numbers of central memory T cells. T cells expanded in these bioreactors were functional, as indicated by killing and producing cytokines and granzyme B. Overall, the work presented here shows that whatever your preferred system is, our GMP ImmunoCult reagents can aid in reliable and consistent T-cell manufacturing in closed bioreactor system. This is crucial for the successful development of life-changing T-cell therapies. I would like to acknowledge the people at STEMCELL who made this work possible. If you'd like to learn more, please visit our website or reach out to your local sales representative. With that, thank you for your attention.